In this chapter, chapter 8, we're going to talk about microbial metabolism. So metabolism refers to all of the various chemical reactions that are occurring at any point in time within a cell. Now, as with a lot of things, we like to categorize things and subdivide them. So there's two different types of metabolic reactions. Catabolic reactions are those that take a larger molecule and you're going to break it down into smaller ones. So you're breaking bonds. Whenever you break a bond, you're going to be releasing energy. So it's also called an exergonic reaction. The reverse of that is an anabolic reaction. You're going to take smaller molecules and connect them together, forming bonds. That's going to require some energy. So you're taking two or three smaller things, connecting them together to make a larger molecule. It's referred to as an endergonic reaction because it does require energy, like I said. Now you may notice that if a catabolic reaction is releasing energy, that's fine. But an anabolic reaction requires energy. Where does it get that energy from? Well, it's going to get the energy from the catabolic reaction that release that energy. So the two have to be connected or tied together. Think of it kind of like a circle that they feed off of each other. It just depends on the needs of the cell, what's going on at any particular time. Along with metabolism, we do classify organisms multiple different ways. And when we're looking at metabolism, we can classify organisms according to their energy source as well as their carbon source. If an organism gets its energy from light, so therefore it's taking that sunlight through photosynthesis, it is breaking it down. It is referred to as a phototroph. If it gets its energy not from the sunlight, but from breaking down chemicals, so it's using chemical energy as a source, it is known as a chemotroph. Then when you look at the source of carbon, if it uses inorganic carbon, remember organic compounds have both carbon and hydrogen, but if it's able to use an inorganic carbon source, such as carbon dioxide, CO2, it's known as an autotroph. If instead it uses organic sources of carbon for its carbon, it is known as a heterotroph. So now you just put these terms together. A photoautotroph uses light energy but inorganic carbon sources. A photoheterotroph uses light energy but organic carbon source. A chemoautotroph will use chemical energy, inorganic carbon source, and then a chemoheterotroph, such as humans, use chemical energy but organic carbon sources. You'll find that microorganisms can fall into all of these different categories. As humans, like I just said, we're chemoautotrophs. But it depends on which bacteria you're looking at, depends on which algae you're looking at as to in which category it's going to fall. Oftentimes in chemistry, we talk about redox reactions. That's an abbreviation for oxidation reduction reactions. What this has to do with is you're transferring electrons. So oftentimes when you're breaking down or building up compounds, you're moving electrons around. Usually you move a pair of them. And so that's what an oxidation reaction is you're referring to. Something gets oxidized, another compound gets reduced. So how do you know which is which? One way to remember it, the way I learned it now, keep in mind I went to school in Lafayette, Louisiana, which is a big oil town, or it was when I was there. So I learned it as the term oil rig. And what that stood for was oxidation is loss, reduction is gain. And you're talking about the electrons. So if something lost some electrons in the reaction, you would say it was oxidized. If something gained electrons, then you say it's reduced. So if something gains electrons, how are you moving these electrons around? And there are electron carriers. These are some very common ones, and in a little bit we'll see some of them again. NAD, when it picks up a pair of electrons, it becomes NADH. FAD, when it picks up a pair of electrons, it becomes FADH2. And then you also have NADP and NADPH. ATP. ATP is energy. ATP stands for adenosine triphosphate. Phosphate bonds are very high energy bonds. And so what happens if you break one of those, triphosphate means it has 3 phosphate bonds. If you break one of those, that's going to release a lot of energy. If you have a lot of excess energy, then you stick a phosphate back on the compound. So this reaction is reversible. You're just constantly moving the phosphate groups off and on, releasing energy, storing energy. Think of it as your ATP is your energy ATM machine. And so when you need energy, like I said, you just take one of these phosphate groups off and it releases the energy. You can do it, whatever work you need to do now. And if you have excess energy, you just stick the phosphate back on and store it there. Enzymes are proteins. Enzymes are a biological catalyst. That means they speed up the rate of the reaction. They do this by lowering what's known as the activation energy. That's how much energy you have to put into a reaction in order for it to occur. The enzyme is not destroyed in the reaction, so therefore it can be reused multiple times. On the enzyme is a site known as the active site. This is where the substrate is going to bind. It's very specific for an enzyme in the substrate. In other words, think of it this way. The active site is similar to a keyhole. In the substrate's the key. So it's very, very specific as to what key can fit into the keyhole. In other words, it's very specific what substrate can bind to the enzyme at that active site. Whenever you have two things binding, such as a substrate binding to the active site of the enzyme, there's always going to be a conformational change. There's always going to be a shape change involved. Now, the enzyme can be reused over and over again. What are some things that can affect the activity level of the enzyme? Well, environmental conditions will play a role. Temperature definitely plays a role. pH can play a role. Sometimes with enzymes you hear cofactors and coenzymes. A cofactor is an inorganic ion that's going to help assist that enzyme do its job. One very common one is iron. They're often a mineral such as iron, magnesium, things like that. Coenzymes are organic molecules that are going to help the enzyme, typically things like vitamins. So when I mentioned that an enzyme lowers the activation energy, the green line seen on this graph is the amount of energy that has to be put into the reaction in order for that reaction to occur. But notice the red dashed line. That is the activation energy that's needed for the reaction to occur. But this is now with the enzyme. So you notice the difference in the peaks right here. This difference, how the amount of energy you have to put in to start that reaction is less. So that's the way an enzyme is going to function. This is just showing what I was saying with you have an active site where the substrate is going to bind. So the active site, the gray area here is your enzyme. The active site is on the enzyme. It's the keyhole. And then here comes the key. Here comes the substrate. It's going to bind. When it fits in there, just perfect, then you have this conformational change and then you release your products in this case. If it doesn't fit in here, it's just going to move on. Nothing will happen. So sometimes I say you need to have a coenzyme or a cofactor, something that helps out. And remember, the difference is just whether it's organic versus inorganic. It's going to typically bind first and then allow the substrate to bind. Sometimes we have inhibitors. Inhibitors are compounds that will either directly or indirectly prevent the reaction from occurring. And how does this happen? If it is a competitive inhibitor, what it's going to do is it is a compound that will compete directly with the substrate for that active site. It can also fit in that keyhole. It's like you have two keys. Who's going to get there first? If the substrate gets there first, then the reaction occurs. But if the competitive inhibitor gets there first, it's taking up the space. There's no room for the substrate to bind. So it directly competes with the substrate for that active site. Now, a non-competitive inhibitor, it's going to compete indirectly because it's not going to bind at the active site. It's going to bind somewhere else on the enzyme. But remember just a bit ago I said whenever you have two things bind, you're always going to see a shape change occurring. So when that non-competitive inhibitor binds at its spot elsewhere, not in the active site, somewhere else on the enzyme, it's still going to cause a shape change. And that shape change now alters the active site. So the substrate can't fit in there anymore because the shape changed. So it still prevents the reaction from occurring. It's just not competing directly with the substrate for the exact same spot. Oftentimes with various metabolic reactions, we see what's known as feedback inhibition. This is actually a very smart way of regulating how the reactions occur. In feedback inhibition, often what happens is you have a series of chemical reactions. One thing you'll learn is nothing ever happens in one step. It's usually multiple steps. And whatever the final product is, that turns around. That's the product you want for whatever the reaction is. That's great. But that product also turns around and acts as an inhibitor, typically at the first step, maybe the second one, but it's going to be more efficient if it acts as an inhibitor at the first step. So you've made this product, it turns around and it shuts down the reaction by acting as an inhibitor. It's very effective at being a type of regulation. Turn it off. So in this picture over here on the left side, we have the competitive inhibition where you have the square here is the competitive inhibitor. It went and it bound right in the active site. So this green substrate, it's not fitting in there. It can't go there. It's kind of like you're in a crowded parking lot and there's only one parking spot left and you see it, you're a couple rows away and you're like, ooh, there's a spot, I'm gonna get that spot. And by the time you go up to the end of the row, turn down the next one and get there, somebody else beat you to it and they got the last spot. It's kind of the same thing like this. Now the non-competitive inhibition, which is shown over here on the right side, you have the inhibitor, but it doesn't bind in the active site. It binds elsewhere. And when it binds there, like I say, it changes the shape for the active site. So now that substrate, it can't fit in here. Excuse me. There is no way that this green half circle is going to fit in this little spot here. It just won't fit. So you're still preventing the reaction from occurring, just indirectly. It's kind of like with the parking lot scenario. You think there's a spot, and by the time you get there, that's when you realize that apparently somebody never learned how to park. There's always that person that instead of parking between the white lines, they park on the white lines, so they're taking up two spots, which now means everybody next to them can't really fit into the spot, and there's no room for you to fit your car in. And you want to say, go back and take driver's ed, because you can't park your car. And this is showing what I mentioned, using feedback inhibition as a means of regulating a particular experiment from occurring. So you're going through, you have your initial product, And like I say, normally it's multi-steps. You get this compound, and then this activates this one, and then the purple activates the blue, and then the blue comes back around, and now you have your final end product, which is this little yellow square, which comes back around here, boom, and it acts as an inhibitor. It's going to shut down the reaction. So now you can't make any more, but that's fine because you've got enough of your product now. Now, why are we talking about all these things on how reactions occur? It's because we're going to look at some very specific reactions. Cellular respiration. This occurs in the majority of cells. It occurs in our human cells. It occurs in a lot of bacteria. It will occur in fungi, so it's kind of a universal reaction that occurs in most organisms. What cellular respiration is referring to is you are taking glucose, which is a carbohydrate, it's a sugar. You require oxygen. You're going to break that glucose down, and you're going to form carbon dioxide, which is CO2. You're going to form water, and you're going to form ATP. A lot of people forget ATP is part of this reaction. That's the whole purpose, is to get energy. This is the most efficient method of producing ATP. It is what we call aerobic because it requires oxygen. Now, not all of the steps require oxygen, but overall it requires oxygen. Cellular respiration is broken down into 3 stages. You have glycolysis, you have the Krebs cycle, and then oxidative phosphorylation, which itself is subdivided into the electron transport chain and chemiosmosis. Glycolysis. We're going to look at all these steps here. Glycolysis. You're taking glucose, which is a 6-carbon compound. It is broken down into 2 pyruvic molecules. No oxygen is required at this step. Oh, and by the way, pyruvate is a 3-carbon compound. You have to initially put in 2 ATPs, but then later you gain or produce 4, so you have a net gain of 2 ATPs and 2 NADHs. NADH is, if you remember, those electron carriers. We have to account for all of those. Now, I know this is a little bit hard to see following your book. First half of glycolysis, that is going to use 2 ATPs. You are going to start, let me change color here. You are going to start with glucose right here, that 6-carbon compound. The next step, you're going to require ATP. You're going to stick a phosphate on the end, and you're going to have glucose 6-phosphate. In chemistry, we number the carbons. So glucose has 6 carbons, so they're numbered 1 through 6. So the way we name it is just tell me you put the phosphate group on the number 6 carbon. Now you're going to shift things around. Glucose and fructose are what are known as isomers. They have the same chemical formula, but structurally they look different. And so it has to do on the very end here as to where the carbon with the double bonded oxygen is. So you go from glucose 6-phosphate to fructose 6-phosphate. Now you're going to have another ATP come in, stick a phosphate on the other end of the chain, so on the number 1 carbon. So now you have fructose 1,6-biscarbon. Fructose 1,6-bisphosphate, also known as fructose 1,6-diphosphate. Different people will name it just a little bit differently. And then what you're going to do is the 6-carbon compound, you're going to break it into 2 3-carbon compounds, and you will end up with glyceraldehyde 3-phosphate. We're only halfway through, okay? Do you have to know the names of the enzymes? No, you do not, but you do need to know the names of the compounds. You need to know when ATP is coming in and ATD is leaving. So everything highlighted here, you need to know those, okay? Now, for the next part of the reaction, we are going to continue on. We're over here. You had the glyceraldehyde 3-phosphate. Now you're going to come in with NAD. It is going to come in, pick up a pair of electrons, leave as NADH. Now, we compare everything to glucose. So remember, we had 2 of these starting glyceraldehyde 3-phosphates. So that's why it's saying you have 2 of these. What you now have, because you had the phosphate come in, you have now 1,3-bisphosphoglycerate. Now you're finally going to take a phosphate group off. One of the phosphates leaves as ATP. This is occurring through both chains. And you end up with 3-phosphoglycerate, meaning the phosphate group's on the number 3 carbon. Now you're going to shift that phosphate to the number 2 position, and it becomes 2-phosphoglycerate. You're going to have a water leave right here. And when you pull off that H2O, you are now left with phosphoenolpyruvate, and then you're going to take that last phosphate off, attach it to ADP, forming ATP, and you are left with pyruvate. So once again, everything that's highlighted is what you need to know. If I were to show you this reaction, could you name the players? Could you name the compounds? Could you name where anything is, what's coming in, what's leaving? Can you do that? If I will be nice and give you a word bank on there. So you now have pyruvate, which is 3-carbon compound. And if you're wondering, why does this take 10 steps to do this? I could just cut it in half. Step 1. You have to remember that these compounds, even though we're drawing them 2D on a flat surface, they're actually 3D. And you have interactions that are occurring. And so once again, my analogy, it's kind of like you're taking a group of 5-year-olds on a field trip on a school bus. Well, you can have all the kids get on and say, okay, 2 kids per seat. And then you kind of go, the teacher is the one who knows, wait a minute, I can't put those 2 boys in the back next to each other because that just spells trouble. So one of you boys, I need to move you up front. Oh, and this little girl, you know what? Last field trip, you threw something out the window. So you can't sit next to the window. So I need you to change places and move over here. Will you stop pulling her hair? You can't be behind her. And so they shift them all around. And it's kind of what's going on. You're adding these phosphates, and I don't like being real close to you, and so I have to shift around, and it ends up taking 10 steps to go from the 6-carbon glucose to the 3-carbon pyruvates. Now, you have those 2 pyruvates. Remember, they're 3 carbons. And by the way, if you noticed in glycolysis, oxygen was not required. We now have to do this little transition reaction because pyruvate cannot feed directly into the Krebs cycle. So what we have to do before we get to the Krebs cycle is we have to convert pyruvate into acetyl-CoA. Acetyl-CoA only has 2 carbons. And so in this transition step, oxygen's not required. You are going to have a net gain of 2 NADHs. And so what happens here is you have the pyruvate, and we have a lot of things going on here. You're going to pull off a pair of electrons, so here's NAD, NADH. You're going to take off a carbon in the form of carbon dioxide. You're also going to have coenzyme A coming in. And what do you end up with now? You end up with acetyl-CoA. That is a 2-carbon compound. That can now feed into the Krebs cycle. Once again, keep track of that NADH because that's going to come up again a little bit later. Now, the Krebs cycle. This is one of those reactions that I guess nobody can agree on. In microbiology, we refer to it as the Krebs cycle, named after the person that discovered it. In most biology courses, they will call it the citric acid cycle. And in chemistry and biochemistry, they will call it the tricarboxylic acid cycle. It's all the same thing. We just give it different names. Now, for this step, just to let you know, oxygen is still not required. However, this reaction will not take place if oxygen is absent. And the reason for that is because you're going to generate a lot of things from this reaction. As you can see, you're going to gain 2 ATPs, 6 NADHs, 2 FADH2s. FADH2s are another electron carrier. It's kind of like you're going to produce all this stuff with no place for it to go, and it's going to build up if you don't have oxygen present. So why waste your time making it? It's kind of like if you, in the area here where we're living in Lufkin, I am fully aware that some people do watch my videos who are not in this area. So Lufkin being 120 miles north of Houston, if you were to go down Highway 59, which is a direct route from Lufkin to Houston, it's a straight shot. Well, if you get halfway down there and suddenly you find out, or you're almost halfway there, say in Livingston you find out there's a really bad accident, whatever, the highway is shut down, or there's flooding or something, you can't get through. Let's say it's an accident. You don't know when the highway is going to be reopened, and you have to get to Houston. Are you just going to sit there and continue driving until you get to Livingston and sit in the traffic jam, not knowing how long it's going to take for it to be cleared up? Or prior to getting to Livingston, maybe you're going through Corrigan, one of these other towns beforehand, and you know that there's a side highway you can take that will cut around. Yes, it's going to be longer, but maybe it'll cut you over to the highway to get you to Beaumont and then Beaumont to Houston. Is it going to add extra time? Yeah. Is it as efficient? No. But will it get you to your end destination? Yes. It's kind of like that. Okay, I'm going through these reactions because I'm trying to make energy. I'm trying to make ATP. But why am I going to go through the Krebs cycle if I'm just going to make a lot of these products, the NADHs, FADH2s? I'm going to generate those, but I can't do anything with it. That's kind of a waste of my time. So do I need oxygen for this for Krebs cycle to occur? No, but if it's not present, I'm not going to go through it, because what I do right after the Krebs cycle, I do need oxygen for. So what is with the Krebs cycle? You had your acetyl-CoA in that transition step that we made from the pyruvate. It is a 2-carbon compound. It is going to combine with this 4-carbon compound called oxaloacetate, and it will form citrate, which is a 6-carbon compound. You're going to have a little bit of readjustment here, and you're going to form isocitrate. Now you're going to have NAD come in. It leaves as NADH. You're going to pull off one of those carbons as CO2. And what are you left with? You're left with alpha-ketoglutarate. Now you're going to have another NAD come in, leave with a pair of electrons as NADH. You are also going to have coenzyme A come in, and you're going to make succinyl-CoA. Now you're going to have removed that coenzyme A. You're going to end up with succinate in this process also. So here's your coenzyme A leaving. You're going to go through this process of taking ADP and forming ATP. It may also involve a side step with GTP and GDP, but you end up with succinate. Now you're going to have your other enzyme carrier, FAD. It's going to leave us FADH2, and you have fumarate here. Excuse me. You have water right here coming in. and you form malate. We still have just 4 carbons here. And then you have NAD come in, take off a pair of electrons, and you're now back to the oxaloacetate that you started with the acetyl-CoA. Now, keep in mind, once again, that we base everything per glucose, which was 6 carbons. But remember, at the end of glycolysis, we had 2 pyruvates, which means you had generation of 2 acetyl-CoAs. So you're going to actually go around, per glucose, you're going to go around this Krebs cycle twice. Okay? Oxidative phosphorylation, which is broken down into the electron transport chain and chemiosmosis. The electron transport chain, this is where your NADH and your FADH2s, those electron carriers, this is where they're dropping off those electrons. And the electrons are going to be passed from one compound to another. And the final thing that receives those electrons is oxygen. That's why oxygen is required. Chemiosmosis is going to be involved. It uses an enzyme, an ATP synthase, to make ATP. In general, for every single NADH that comes in, and if you count them all, there's going to be 10 of them, it will make, for each NADH, it generates 3 ATPs. For the FADH2s, it will only generate 2 ATPs. And there were 2 of those, so that'd be a total of 4. So, why is there a difference? Well, right here we're going to look. This is where the NADH comes in, it drops off its pair of electrons, and they're going to be just passed along between all these various protons in here, or proteins, and then finally over here, look, right there, oxygen is the last thing to receive it. FADH2 comes in a little bit further down, so the electrons aren't passed quite as far. That's why they only generate 2 instead of 3 ATPs. And as you can see, when you drop out the pair of electrons, this converts back to the NAD or the FAD, which can go back and be reused again. So you started off with glucose. You had the requirement for oxygen here. That's the final electron acceptor. You saw throughout the process that as we pulled off carbons, they left as CO2, which is carbon dioxide. Here is your water that you're forming, which is one of the other products. And then finally over here, every time you pass a pair of electrons down this chain, you have hydrogens that are moving across the membrane through these proteins. You end up with a high concentration of hydrogen ions on this side. It's going to move through this enzyme down here, and those as the hydrogen ions come through, they're going to help generate, basically be producing ATP, which is the final product also. Remember, cellular respiration. Glucose plus oxygen is going to give you carbon dioxide plus water plus energy in the form of ATP. Most of the ATP is being produced by the electron transport chain in the oxidative phosphorylation step. So if you look at all of it, in glycolysis, in this step, you generated 2 NADHs. You had a net gain of 2 ATPs. So over here, this transition state, converting the pyruvates to the acetyl-CoA, you generated 2 NADHs. The Krebs cycle, you generated, if you count them up, remember you went around that cycle twice, so you have 6 NADHs and you have 2 FADH2s. So what is your total? Like I just said, you had 10 NADHs and you had 2 FADH2s. And so how many ATPs, oh, over here I forgot to notice, you also on the Krebs cycle had 2 ATPs generated there. How many ATPs did you generate total? Well, you have the 4 ATPs, 2 from glycolysis, 2 from Krebs cycle. If you count up you have 10 NADHs, each of them generates 3, so that's 30. And then the 2 FADHs over here, you generate 2 for each of them. And so that is a total of 34 just from the oxidative phosphorylation step. For prokaryotic cells, that is a total of 38 ATPs per glucose. That's quite a bit. In eukaryotic cells, the number is only going to be 36 total ATPs generated, and that is because in a eukaryotic cell, glycolysis occurs in the cytoplasm, but then that pyruvate has to be transported before the transition step. It has to be transported across the mitochondria. Passing that pyruvate across the mitochondrial membrane is going to cost you 2 ATPs. And everything else occurs inside the mitochondria. Prokaryotic cells don't have a mitochondria. This is just occurring with proteins along the membrane of the cell membrane. So what happens if oxygen is not present? Well, you still need to have ATP to run various reactions. And so in this case, one of the common things that will happen is that the cell will go through what is known as fermentation process. This is anaerobic. It means if something is anaerobic, it's without oxygen. Now, in this case, only 2 ATPs are going to be generated. Not nearly as efficient. You're talking 2 versus 38, but at least it's enough to keep you going. When you carry out fermentation, you can have a couple different products depending on which organism you're talking about, depending on the substrate that's present. Typically with fermentation, you can end up with what we call either acid fermentation or alcohol fermentation. It's like I say, it's going to depend on the species. Human cells do acid fermentations. We produce lactic acid. When you're talking about microorganisms, it just depends on the species that you're talking about. One thing, we often will look at fermentation and how a microorganism is able to utilize a particular, say, sugar. Is it able to ferment it? What's the product? Does it produce acid? And once again, this helps us with identification steps. If something is said to be homolactic fermentation, it means that only lactic acid is produced. If it's heterolactic fermentation, it means not only is lactic acid produced, but also the gas, carbon dioxide. And there's a way that we can measure that gas. So we do make a distinction between, was there just acid or was it acid and gas? Once again, different species with different substrates may give different products. And that, once again, those differences are helping you to identify the organism. Lipid and protein metabolism, just so you know, there's a lot of different intermediates can leave anywhere along the reaction. So most of the reactions will eventually connect to either glycolysis or the Krebs cycle. So you don't have to start at the beginning. A lot of reactions end up producing glyceraldehyde 3-phosphate, which is in the middle of glycolysis, and so that's where it can feed into, and that's fine. Lipases are enzymes that break down lipids. Proteases are enzymes that will break down proteins. Biogeochemical cycles, what we're talking about here is nutrient cycling, the recycling of things like carbon, nitrogen, sulfur, phosphates, all of that. Microorganisms play a significant role in nutrient cycling. Bioremediation is using microorganisms to remove or break down pollutants that are either in soil or water. Xenobiotics are man-made compounds in the environment. They're in higher concentrations than what would naturally be found there. Typically, you're talking about pollutants. They shouldn't be there in that concentration. This is just showing an example of the carbon cycle, of how things are growing. You have carbon tied up in plants and excuse me, different vegetation. you have production of CO2, and then how as plants die, that carbon gets back in the soil, etc., and just how everything is interconnected. Same thing with the nitrogen cycle. A lot of things are tied up in nitrogen in plants and then the soil. And so everything, once again, just cycles around. Microorganisms play a huge role in all forms of the nutrient cycling. And this is showing with the sulfur cycling as well. The main thing from this chapter that I want you to be familiar with is just overall how reactions occur, catabolic versus anabolic. But by all means, know cellular respiration. Know all the 3 stages of it in detail. Know the various compounds, what's coming in, what's leaving. If I were to give you a copy of the reaction with no names, and then I give you a separate page with a word bank, can you fill it in? And hopefully your answer is going to be yes, or at least by the time of the test, yes, you will be able to do that. You'll be able to fill in where all the ATPs and ADPs and NAD and NADHs, FADH, FADH2s, as well as the glucose, the glyceraldehyde 3-phosphate, fructose 1,6-bisphosphate, the fumarate, malate, all of those different ones. Can you fill them in? One of the reasons I'm kind of picky on this is, number one, I'm a physiologist, so I find this area fascinating. The main reason for you is because these reactions occur not just with microorganisms, but also with your cells. It's universal. All living cells can carry this out. And it's such a basic, fundamental process in physiology that it's so important that you understand how it works. You understand the steps of it. And so that's why you have to know it.